हेलो फ्रेंड्स। स्टार्ट करते हैं। हमारा यूनिट 5 है ह्यूमन फिजियोलॉजी। द रिडक्शनिस्ट अप्रोच टू स्टडी ऑफ लाइफ फॉर्म्स रिजल्टेड इन इंक्रीजिंग द यूज ऑफ फिजिको केमिकल कॉन्सेप्ट्स एंड टेक्निक्स. Majority of these studies employed either serving tissue model and or state way cell free systems. An explosion of knowledge resulted in molecular biology, molecular physiology because became almost synonyms with biochemistry and for biophysics. However, it is now being increasingly realized that molecular approach would reveal the truth about biological processes or living phenomena systems. Biology makes us believe that all living components of the system under study, regulatory network of molecules, supra molecular assemblies, cells, the tissues, organisms, and indeed populations and communities each create emergent properties. In the chapter under this unit, major human physiological processes like exchange of gases, blood circulation, locomotion, and movement are described in cellular and molecular terms. The last two chapters point to the coordination and regulation of body events at the organization level. Alfonso Corti, an Italian anatomist, was born in 1822. Corti began his scientific career studying the cardiovascular system of reptiles. Later, he turned his attention to the mammalian auditory system. In 1851, he published a paper describing a structure located on the basilar membrane of the cochlea containing hair cells that convert sound vibrations into nerve impulses. The origin of Corti. He died in the year 1888. फोर्थ नंबर का चैप्टर है ब्रीदिंग एंड एक्सचेंज ऑफ गैसेस. एज यू हैव रीड earlier, oxygen O2 is utilized by the organisms to indirectly break down simple molecules like glucose, amino acids, fatty acids, etc. to derive energy to perform various activities. Carbon dioxide CO2, which is harmful, is also released during the above catabolic reactions. It is therefore very evident that O2 has to be continuously provided to the cells, and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following sections of this chapter. Respiratory organs, mechanisms of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, ciliates, flatworms, etc. exchange O2 with CO2, carbon dioxide, the simple diffusion over their entire body surface. Earthworms use their moist cuticle, and insects have network of tubes, tracheal tubes, to transport atmospheric air within the body. Special vascularized structures called gills, branchial respiration are used by most of the aquatic arthropods and molluscs, whereas vascularized bags called lungs, pulmonary respiration are used by the terrestrial forms for the exchange of gases among vertebrates. Fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin. The cutaneous respiration also. Human respiratory system. We have a pair of external nostrils opening out above the upper line. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage of for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis can be covered by a thin plastic cartilaginous flap called epiglottis to present to prevent the entry of food into the larynx. Trachea is a straight tube. Trachea is a straight tube extending up to the the thoracic cavity, which divides at the level of fifth thoracic vertebra into a right and left primary bronchi. Each bronchi undergoes repeated divisions to form the secondary and tertiary bronchi and bronchioles ending up in very thin terminal bronchioles and the trachea, primary, secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structure called alveoli. The branching network of bronchi, bronchioles, and alveoli comprise the lungs. Figure 14.1. We have two lungs which are covered by a double layered pleura with pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts form the respiratory and exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from foreign particles, humidifies, and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an air-tight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of the lungs in thorax is such that any change in the volume of a thoracic cavity will be essential will be reflected in the lung pulmonary cavity. Such an arrangement is essential for breathing as we cannot directly alter the pulmonary volume. Respiration involves the following steps. First, breathing or pulmonary ventilation, by which atmospheric air is drawn in or to carbon dioxide rich alveolar air is released out. Second, diffusion of gases O2, CO2 across the alveolar membrane. Third, by the blood transport of gases by the blood, diffusion of O2 and CO2 between blood and tissues. Now, fourth, diffusion of O2 and CO2 between blood and tissues. Fifth, utilization of O2 by the cells of catabolic reactions and resultant release of CO2, cellular respiration, as did in the chapter 12, mechanism of breathing. Breathing involves two steps: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by breathing. Or pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs intrapulmonary pressure is less than the atmospheric pressure. That is, there is a negative pressure in place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the anteroposterior axis. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decrease the infra pulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. That is inspiration. Figure 14.2a, relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their volume, and thereby the pulmonary volume, and this leads to an increase in infra pulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, and that is expiration. Figure 14.2b. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, the healthy human breathes 12 to 10 12 to 16 times per minute, and the volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. Respiratory volumes and capacities. Tidal volumes, TV. Volume of air inspired or expired during the normal respiration, it is approximately 500 milliliter. That is, a healthy man can inspire and expire approximately 6000 to 8000 milliliter of air per minute. Inspiratory reserve volume, IRV, additional volume of air a person can inspire by a forcible inspiration, and this average 2500 milliliter to 3000 milliliter. Expiratory reserve volume, ERV, additional volume of air a person can expire by a forcible expiration. This average 1000 milliliter to 1100 milliliter. Residual volume, RV, volume of air remaining in the lungs even after a forcible expiration. This averages 1100 milliliter to 1200 milliliter. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity, IC, total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume. TV plus IRV. Expiratory capacity EC. Total volume of air now person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume TV plus ERV. Functional residual capacity volume of air that will remain in the lungs after a normal expiration. This includes EV, ERV plus RV. Vital capacity VC, the maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV, and IRV, or the maximum volume of air a person can breathe or after a forced out of or after a forced inspiration. Total lung capacity TLC, total volume of air accumulated in the lungs at the end of the at the end of forced inspiration. This includes RV, ERV, TV, and IRV, or vital capacity plus residual volume. Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure upon concentration gradient, solubility of the gases, as well as the thickness of the membranes. Membrane gradient, solubility of the gases, as well as the thickness of the membrane involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called a partial pressure, and is represented as O2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in the atmospheric air and the two sites of diffusion are given in table 14.1 and in figure 14.3. The data given in the table clearly indicates the concentration gradient for oxygen from alveoli to blood and blood to tissues. Similarly, a gradient is present for CO2 in the opposite direction, that is from tissues to blood and blood to alveoli, as the solubility of CO2 is 20 to 25 times higher than that of CO2 O2. The amount of CO2 that can diffuse through the diffusion membrane per unit decrease difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers, figure 14.4, namely the thin, the thin, the thin squamous epithelium of alveoli, the endothelium of substance composed of thin basement membrane supporting the squamous epithelium, and the basement membrane surrounding the single layer endothelial cells of capillaries. In between them, however, its total thickness is much less than a millimeter, and therefore all the factors in our body are favorable for diffusion of O2 from alveoli to tissues, and that of CO2 from tissue to alveoli. Transport of gases. Blood is the medium of transport of O2 and CO2. About 97% of O2 is transported by RBCs in the blood. The remaining 3% of O2 is carried in a dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried as breakdown, um, uh, carried as carried as bicarbonate. About 7% of CO2 is carried in a dissolved state through plasma. Transport of oxygen. Hemoglobin is a red colored ion containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2 oxygen. A binding of oxygen with the hemoglobin is primarily related to partial pressure of O2, partial pressure of CO2, carbon dioxide, hydrogen ion concentration, and temperature are the other factors which can therefore with this binding, the sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. This curve is called the oxygen dissociation curve, figure 14.5, and is highly useful in studying the high effect of factors like PCO2, H positive concentration, etc., on binding of O2 with hemoglobin in the alveoli, where there is higher PO2, low PCO2, lesser H positive concentration, and lower temperature. The factors are factors of all favorable for the formation of where low PO2, high PCO2, high H positive concentration, and higher temperature exist. The conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets oxygenated blood can deliver around 5 milliliter of O2 to the tissues under normal physiological conditions. 14.4.2 transport of carbon dioxide. CO2 is carried by hemoglobin as carbaminohemoglobin, about 20 to 25%. This binding is related to the partial pressure of CO2. PO2 is a major factor which would affect this binding when PCO2 is high and PO2 is low, as in the tissues. More binding of carbon dioxide occurs, whereas when the PCO2 is low and PO2 is high, as in the alveoli. Dissociation of CO2 from carbaminohemoglobin takes place, that is CO2 which is bound to hemoglobin from the tissues delivered at the alveoli. RBCs contain a very high concentration of the enzyme carbonic anhydrase and minute quantities of the same is present in the plasma too. Now, this enzyme facilitates the following reaction in both the directions. CO2 plus H2O water in presence of carbonic anhydrase then gives the H2CO3. In presence of carbonic anhydrase then gives the HCO3 negative plus H positive. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBCs, and plasma and forms HCO3 negative and H positive at the alveolar site where PCO2 is low. The reaction proceeds in CO2 trapped is bicarbonate at the tissue level and transported to the alveoli is released out as CO2, figure 14.4. Every 100 milliliter of deoxygenated blood delivers approximately 4 milliliter of CO2 to the alveoli. The regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural the system, a specialized center for present in the medulla region of the brain called the respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called the pneumotaxic center can moderate the functions of the respiratory rhythm center neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. The chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate this center, which in turn can signal the rhythm center to make the necessary adjustment in the respiratory process, by which these substances can be eliminated. Receptors associated with the aortic arc and carotid artery also can recognize changes in CO2 and H positive concentration and send necessary signals to the rhythm center for remedial actions. The role of oxygen is the regulation of respiratory rhythms is quite insignificant. Disorders of respiratory system, asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. Now, one of the major causes of this is cigarette smoking. Occupational respiratory disorders in certain industries, especially those involving grinding of or stone breaking. So much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation, leading to fibrosis, proliferation of fibrous tissues, and thus causing serious lung damage. Workers in such industries should wear protective masks. Breathing and exchange of gases. Start करते summary. Cells utilize oxygen for metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cells and for the removal of carbon dioxide from there. Now we have a well developed respiratory system comprising two lungs and associated near passages. To perform this function, the first step in respiration is breathing, by which atmospheric air is taken in the inspiration and the alveolar air is released out. The expiration, exchange of O2 and CO2 between deoxygenated blood and alveolar transport of these gases throughout the body by blood. Exchange of O2 and CO2 between the oxygenated blood and tissues, and utilization of O2 by the cells. Cellular respiration are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles. Intercostals and diaphragm volumes of air involved and these activities can be estimated with the help of a spirometer and are of clinical significance. Exchange of O2 and CO2 at the alveoli and tissues occur by diffusion. The rate of diffusion is dependent on the partial pressure gradient. Gradients of O2, PO2 and CO2, PCO2. Our body facilitates diffusion of O2 from the alveoli to the deoxygenated blood, as well as from the oxygenated blood to the tissues. The factors are favorable for the diffusion of CO2 in the opposition direction, that is from tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin in the alveoli, where PO2 is higher. O2 gets bound to hemoglobin, which is easily dissociated at the tissues, where PCO2 is low and PCO2 and H positive concentration are high. Nearly 70% carbonic acid anhydrase, 20 to 25% carbon dioxide, is carried by hemoglobin as carbaminohemoglobin in the tissues where PCO2 is high. It gets bound to blood, whereas in the alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. Respiratory rhythm is maintained by the respiratory center in the medulla regions of brain, the pneumotaxic center in the pons region of the brain, and chemosensitive area in the medulla can alter respiratory mechanism. सो यहाँ पर समरी ख़त्म होती है. सो हमने पहले से देखा ब्रीदिंग एंड एक्सचेंज ऑफ गैसेस में रेस्पिरेटरी ऑर्गन्स, ह्यूमन रेस्पिरेटरी सिस्टम, mechanism of breathing, respiratory volumes and capacities, exchange of gases, transport of gases, transport of oxygen, transport of carbon dioxide, regulation of respiration, disorders of respiratory system, then summary. So like, share and subscribe. Thank you so much for watching.